what I want to present in this unit together with my colleague Vladimir Meglic. We want to discuss the development of these new breeding populations with improved performances under low input and abiotic stresses and other conditions, and their role or possible role as starting materials for breeding programs or for commercializing new varieties. So following the previous ideas discussed in previous modules of the course, using hybridization to incorporate the variability in breeding studies, I am going to give a hint of new experimental populations as a source of this new diversity needed for breeding purposes. So we are going to talk about the type of experimental populations that are currently used in breeding basic experiments. And also I will explain some examples of the use of these populations for understanding the genetic basis of some agronomic important traits in Solanaceae species like tomato and eggplant. And later, Dr. Vladimir Meglic will also speak about the use of this type of populations in other important crops like cereal families and cucurbitacea. So, first of all, I think it is necessary to have in mind that traditional breeding populations, although they have been used during centuries in breeding, and mass selection has been performed, pure line selection during centuries, also pedigree and backcross to integrate some interesting traits into let's say a commercial variety. Although these methods has been widely used for decades, for centuries, these methods also present some disadvantages that we need to have in mind, to take into account now that we know more about how the breeding process works and what do we need to do future breeding programs. For you to understand, normally these populations they usually include only two parental lines, narrowing the genetic diversity of the crop. That means that if we use this type of populations during a lot of time, that will decrease significantly the capacity of future breeders to do breeding, to do improvement of the crop, because we are losing this diversity, this variability. So normally they narrow genetic diversity. They are also phenotype based. That means that when we are selecting through the phenotype, we can drag undesirable traits that are hidden in the genome into the descendants. Even we do not see them in a generation, maybe through intercross, in the next generation, they can appear, that negative phenotypes. They are normally not precise, and they are less cost-effective than modern breeding populations or modern breeding techniques. They are also slow. They take more than 10 years for commercializing a new variety into the market, and they are good for or effective for simple or qualitative traits, depending on just one or few genes, but they are not that useful for quantitative of complex traits like yield. Even a lot of commercial varieties have been gone out into the market with improved yield, for example. New methods have been more efficient for these purposes. So let's talk a little bit about new types of populations that can be very useful also to breeding purposes and to establishing new breeding methods and new commercial varieties. As I was saying, these new breeding populations has been proposed to gather many genetic and phenotypic resources and overall avoid the genetic loss that these traditional breeding methods were causing. So the first example of new breeding populations would be the Near-Isogenic Lines. These Near-Isogenic Lines contains only a simple introgression of one variety into the genome of our interesting crop. That means that we only have one portion of a variety into the genetic background of another variety. Maybe we want, an example, maybe we want to add only genes causing resistance to an illness into a commercial variety, for example. So one example of NILs, of this type of near-isogenic lines, would be the introgression line population. This is the example you see on a screen. On the right, you see how this population is developed. Basically, this population is obtained after one first cross, and after one first cross, the F1 have half and half the genome of both the parentals. But we want to maintain the genome of the commercial variety, let's say, and then we back cross to the commercial variety and select in each back cross the lines that contain the phenotypic trait and the genetic associated marker that we want. For example, I want to introgress resistance to, let's say, a virus. We do the first intercross, and the F1 we back cross to the commercial variety, which did not have first the virus resistance. Let's start again. I have a commercial variety which is not resistant to the virus, and let's say a traditional variety that is resistant to the virus. We do the first intercross, and then we back cross to the commercial variety, but selecting the individuals that have this resistance to the virus. Then after a lot of backcrossing, what we have is the exact genome of the commercial variety with all the good traits we are selecting of the commercial variety, let's say fruit quality, yield, adaptation to the organic conditions, whatever. And then we also select these individuals which also have the resistance to the virus. So this is the idea of the NIL. So in this example, the introgression line populations in eggplant, what they did is select phenotypically and also with markers, with mass marker-assisted selection, as you have seen in the previous unit, to obtain a homogeneous commercial variety but the idea of this population was not only to have this one resistance, for example. What they developed is a population in which I have a commercial line, and then I have represented the genome of a traditional or a wild species into the genome of the commercial line to see which region, which portion, what the genes of these regions, what did they do? Okay, so each line in the ILS population has a different portion of the traditional variety. So with all the introgression lines, we have represented by portions all the donor genome into the commercial genome. Comparing each line, we can see the differences in the phenotypes, for example, different adaptations, different resistances, different tolerances, different fruit quality traits, different agronomic traits, and see which region of the genome is responsible for this trait. So we can not only associate traits to genomic regions, but also we can develop breeding materials for commercializing with a high added value. Another types of new population in breeding are the recombinant inbred lines. So these type of populations exploit the maximum recombination frequency within the existing lines. So as you can see in the image, basically we do a single or multiple cross, just focus on a single one, and then we self this F1 to obtain an F2 with more recombination events, and then we can even self more this F2 and fix through selfing to obtain a collection of lines which would be the RILs population. Each one of these lines is one RIL, one recombinant inbred line, which is a mosaic of the parental genomes. That means that they contain different regions and different combinations of the genomes of the the parental lines, and also are fixed through selfing, which means that they are homozygous and they can be studied, is further studied for gene association with traits, or even selection for interesting improved traits. So the idea is both parental genome are represented in the RILs, but with different combinations of the genes and the alleles. So we can know the effect of each genomic region, basically comparing different lines, comparing the phenotypes, and comparing the genomic regions that are causing these differences. So these RILs can come from a single intercross, or as we will talk in the previous module, they can be coming for multiple parental crosses, which would be called a multi-parent population, and I'll explain it later. So RILs have been developed in a lot of crops. For example, recombinant inbred lines for eggplant have been developed, and you can see in the image that from two parental lines, the F1 was an admixture phenotypically of both parentals, but the real population, you see each eggplant is coming from a different plant, from a different line, a different recombination line, and you can see the very high diversity we have reached from two parentals. So the combinations of the genomes of this parental is very high, and that helps us a lot. For instance, we have here that all these RILs have been tested for improved fruit nutritional quality. So what we can do now is relate which are the plants, which are the RILs with a better nutritional quality, and use them as starters materials for commercializing a new breeding line. Or we can also associate this phenotype, this improved phenotype, with the genes that are causing them. Another RIL population in eggplant, you see there's a lot of work in this matter. So in another RIL population, they have screened for resistance to Ralstonia solanacearum with different strains, and they have also observed a lot of diversity. So you see, it's a useful breeding population for not only basic interest, like not only providing or knowing new genetic information, but also as useful materials for new commercial lines. So now it's time to start to focus in a different type of populations. So I'm now talking about multi-parent populations. Multi-parent populations are based in that we need to include more than two parental lines in the first intercross, and they are useful because we aggregate more variability and we capture additional genetic recombination of many interesting genotypes. So the basic objectives of this type of population, the reason we add that much genomic information, that much genetic variability, is that we can introgress in interesting commercial varieties more than one interesting trait, and avoiding that genetic loss that traditional breeding methods is causing. And we also can obtain genetic information of the traits, relating these genes to complex agronomic traits, to go further into plant evaluation, to ease our way to analyze the lines and select the lines. Basically, the idea of these multi-parent populations is that when we do an intercross, a first intercross, we also do the recombination of the genomes of the parental lines into the descendants, let's say. So when we increase this recombination, we are forcing these genomes to break into shorter pieces of the genome. Instead of having this largest amount of one genome and largest amount of the other parental genome, we are including little pieces of each genome combined, which is very interesting. So the idea is that when we break these genomes, these pieces of genomes into little pieces, we are able to study proximal genes, which would be normally inherited together. We are able to study them when they are inherited separately. So we can study them independently. I think someone's talking, or it's just okay. So the idea is that when we are breaking these species, which we call the linkage disequilibrium, the distances between independent genes, we reduce also the population structure, we can increase mapping resolution, and we can increase the QTL identification. Basically, we are able to ensure that one candidate gene is causing one determinant phenotype. And it's easy for us in very complex traits. So the most popular design of this multi-parent population is the multi-parent advanced generation intercross, or what we call a magic population. These populations are based on the intercross of more than four founder lines, typically 4, 8, or 16 founder lines, but even 24 or 36 in some cases I read, followed by a funnel breeding design. Basically, what they do is intercrossing parents F1 hybrid and F2 hybrid into pairs, obtaining a base generation with recombination of all the 8 genome of the parental ones, of the parental lines. And then these individuals with a high recombination of these parentals become self until they become RILs, which are just the final result of a magic population is a collection of lines of RILs that are recombined different. Each line is a different recombination, a different mosaic of the 8 parental genomes, and display different phenotypes, different combinations of phenotypes, and different combinations of genome features. So they are very interesting tools for associating gene to trait, but also are very interesting to start maybe a breeding for complex traits with a lot of genes depending on the trait. So the good thing about this type of populations is that they are fixed in the last selfing population, in this last selfing generation. Sorry, after 5 or 7 selfing generations, they are fixed. They are homozygous for all the genes. So it's very interesting because it's what we call an eternal population. They can be maintained by selfing eternally, forever. Magic populations have been developed in very economically important crops like tomato and eggplant, or cereals like maize, wheat, sorghum, or rice, and legumes like chickpea or cowpea. And the study of these populations have resulted in a lot of new genes or genetic regions related to complex traits. This information is now being used to improve crop quality and yield, and also adding resistance and tolerance to these two different abiotic stresses during these breeding processes. Now I'm going to give you some examples of magic populations and their use. In our laboratory in Valencia, we are currently developing the first magic population in capsicum developed in the world. It's a magic population of different varieties of pepper, and we have found that this population embed a great variability of the founder lines. We have increased the recombination rate that is normally observed for pepper. We also reduce the population structure that may be normal in other breeding populations, and we have found that after 5 selfing populations, we have reached a high homozygosity, and then that means that this next generation could be used for analyzing complex traits like adaptation to abiotic stresses, adaptation to low input or organic condition, or even finding relations with the genome and the traits like yield or fruit quality, or even improved taste. So, however, we performed a preliminary analysis in the third selfing generations to see how the populations was evolving. So we used the morphological descriptors for plant, leaf, flower, and fruit And we observe a high recombination of the phenotypes, uprising new combinations of the parental phenotypes haven't observed before, new phenotypes for qualitative traits that were not observed in the parental lines, and also heterosis in quantitative traits like fruit weight, and plant height, if I'm not mistaken right now. If you agree, I will answer the questions after the presentation, okay? I will take a look at the chat and answer you later. So, for example, in this population, we have observed that the lines embed all the variability of the parental lines, both phenotypically and also genotypically, including variability coming from all the parental lines, as we can see in the PCA, where we have all the parental lines more or less divided, and a cluster of dots with all the S3, the third self-generation lines. Is diverse. They embed all the parental variability, and even you can see that some points are very far from the first cloud. So it's interesting because some lines are showing heterosis, different characteristics from the parental lines. So in the 5th generation, we are supposed to study more complex traits, more adaptation traits, and fruit quality traits. Moreover, in the 3rd generation, we tried and we associated some traits related to fruit quality, like this. This is fruit color in maturity, with some genomic regions, which is very interesting because with the result, we are trying to relate genes with the trait for fruit quality, with some traits of fruit quality, which is interesting because in that case, we can develop markers to select breeding varieties with improved fruit traits. Another example is an eggplant magic population, which was published 3 years ago in the 2022. And the RILs resulting from this population, the 6th selfing population of all the lines of the magic fixed, have been used, tested, and evaluated for a lot of traits. For example, a collection of these RILs have been tested for water deficient conditions with promising results, which means that some of these varieties can be starting materials for a breeding program or even for commercializing new eggplant varieties tolerant for low water input conditions. They have also analyzed in this RIL population the fruit quality, for example, the color of the peel, and they have found a genomic region and the gene and the mutation of the transcription factors which cause a different pigmentation of the peel. Normally, we won't commercialize purple eggplants, but they discovered that a mutation on this transcription factor caused the peel to become greener, and this devaluate its value. So it's interesting to be able to have these tools to be able to say easily which lines have more fruit quality than others, or a better fruit quality. A lot of populations have been developed in tomato. This population based on an 8 parental first intercross. With this population, they have associated some QTLs to fruit quality traits like color, sugar accumulation, a lot of acid accumulation. So with this magic population in tomato, they have been able to associate QTLs to fruit quality, which is very interesting, to improve quality or flavor in tomato, which has been a concern in the last years for tomato producers. Even very complex magic populations have also been developed with more than 8 parentals, and the idea is accumulating more diversity and creating new combinations of the genomes, which is just accumulating new diversity, and be able to evaluate and screen outstanding performances, outstanding lines, under difficult conditions. For example, this magic population has been tested under drought conditions, which would lead to the selection of better lines for commercializing and cultivation in arid. So just to finish this part, I just wanted to reinforce the idea that this type of descendants populations encompasses a lot of genetic variability, and they are homogeneous and fixated lines, so they might be tested for a lot of traits, also complex traits and traits related to adaptation to different conditions, and also the resultant population of RILs of these new breeding populations can be explored for a better performance under organic and low input conditions. So now it's my turn of Dr. Vladimir Meglic. I silence my mic and I will answer your questions by chat.
Put my mic on. Thank you very much. Good afternoon or good evening to all of you. So we have still a little bit to go today. And I will continue with the, let's say, the magic populations, or with the evolutionary populations that were part of the EcoBeat project that finished in the spring of last year. And this project dealt with the breeding of the field crops, potatoes, wheat, buckwheat, and soybeans. And within the project, we have decided that we will deal as well with the evolutionary populations, with the magic populations, predominantly in wheat. And we have set up, let's say, some of the questions that we need to, let's say, deal with, or problems that we would like to answer. And hopefully I won't bore you too much, or I won't repeat too much, but we have said that there are only a limited number of varieties that are adapted to the environmental variability of low input or organic farming. And genetically homogeneous varieties are more susceptible to spatiotemporal changes in biotic or abiotic stresses, and therefore they have limited adaptability to local growing conditions. And another question or debate was on the environmental variability that often affects crop yields more under low input and organic conditions than in conventional agriculture. So we have said that solutions, or one of the solutions, would be to grow genetically diverse populations that could improve tolerance to a wide range of environmental stresses and different growing conditions, and to allow them to evolve and adapt to changing conditions. So that means even if we have different farmers within a macro region, so then their micro environment could contribute to adaptation of those populations in their specific fields. So these CCPs, or composite cross populations, are populations of segregating individuals that has already been, let's say, discussed quite a lot, that are crossed with each other to produce heterogeneous and diverse material. And this is an example of the evolutionary breeding. And many breeders and researchers have developed and tested heterogeneous populations over the past 2 decades, and we have seen quite a lot of examples already within our series of presentations in last couple of weeks and today. And in EcoBreed, there was one Hungarian variety, Martin Lasser Elite CCP, that was produced by crossing 7 different parents, and was tested in organic field trials in 2020 and 21. So within the project, and shows stable grain yields and high protein content. So we are really glad that as well these CCPs are competitive, let's say, to other populations or the varieties. And when we compare that to variety couple, which is popular with organic farmers and growers, our CCP showed a significantly higher protein content with a similar yield. So this is as well one thing that could contribute to even well-being of farmers, organic farmers, using these kind of populations. Next Slide, please. So here's an example what we have further done in our as an EcoBreed example. Magic populations are created by several generations of intercrossing among multiple founder lines, and those multiple founders contribute more allelic diversity, and multiple cycles of intercrossing give greater opportunities for recombination. So this was really well described in a previous talk. And such multiparental populations are nowadays widely used for the mapping of genes or QTLs, and may also be used for the creation of heterogeneous material for on-farm selection. So this is as well one thing that we were really keen on, to offer our populations to the farmers to make a further selection for their own, let's say, farm environment. Next slide, please. So as well, we have approached and made another set of crosses. We have produced 2 8-parent magic populations that were developed for further selection of either pure line breeding lines or development of organic heterogeneous material under the organic conditions. So the first process started pretty much at the beginning of the project, and the final cross was done in 2021. And we have developed 2 populations that were multiplied then in 22 and 23 as bulks in Hungary and in Austria as ear-to-row progenies. And then that material was further distributed as bulks to partners in Germany, Czechia, Slovakia, and Slovenia. So we had 4 locations where we have tested that material. And below we have a description of varieties that formed the early panel and the late panel. So I'm not gonna read out all the varieties, but those are all the varieties, either the established and popular varieties with the farmers, or already new varieties that were just being developed by the breeders that were partners in the EcoBreed project. So below you can see the table where we have compared the early and the late magic populations on 4 locations, and where we see that the values for BBCH at the 24th of May and the plant height. So we see that there are differences between those 2 populations, which in the continuations will give us as well the opportunity to do a further either breeding or to further mapping of the traits that we would like to look at. So that's how it was already described by Neus. Next slide, please. So I'm going now, let's say, backwards. Neus was developing her story, and we'll go back to the introgression lines to NILs and RILs in different crops. So first, I would like to look at the example that on the introgression lines that I was as well working on when postdocing at UC Davis, and looking at the cross between Solanum lycopersicoides and that was intercrossed into the, or introgressed into the tomato background. And the advantages of Solanum lycopersicoides, we can, there are several and really, let's say, exciting ones. It's the perennial, so like a bush with hard fruits. It's found really at high elevations. It's self-incompatible, but is a cross-pollinating species, has the same number of chromosomes as tomato, and that's why it's possible to cross with tomato. But unfortunately, most of the crosses produced are male sterile and incompatible. It has as well resistance to abiotic stress, low temperature, frost, and drought. It has tolerance to mechanical damage. It has really firm fruits. And as well to the, when we're looking at the resistance to biotic stresses, it's tolerant or resistant to several viruses, bacteria, fungi, and insects. Next slide, please. So this was the hybridization scheme that was developed by Charlie Rick. And first, he was making, let's say, hundreds of different crosses to finally found a, let's say, population that yielded a bigger amount of pollen. And that population was then used for the back crossing into the fresh tomato background. Fortunately, yeah, from that one male fertile, that the whole story started when then with the series of back crosses, selfings as well back crosses, those introgression lines were developed. Next slide, please. And as it was already discussed and shown for eggplant, this was done, let's say, in a similar way, where we have tried to introgress and follow all the small pieces of the chromosome of the chromosome in each of the Solan Lycopersicoides into the tomato genome, and trying to address each chromosome. So in the background, you see the, let's say, the big picture where we have introgressed the whole Solan Lycopersicoides genome into the tomato background. And on the foreground, on the left-hand side, you see the example of chromosome 2, where we have in white homozygous segments of the Lycopersicoides dark tomato genome. That was done with the tools at that time, and I was using a lot of, let's say, RFLPs. Now these populations are used in a further breeding and further, let's say, dissecting the Lycopersicoides genome and the traits that genome is contributing to the new varieties and populations. Next slide, please. Next example is the example of cucumbers, where while I was in University of Wisconsin, we were mapping the cucumber genome. And we were looking first at the current plant times that were monoecious, had unilateral branching, set fruit inhibition, and indeterminate habit. And we have kind of looked at the future or desired ideal type with the plants that would be gynoecious, would have multiple lateral branching, sequential fruit setting, and would be determinate. Next slide, please. For that, we have chosen two experimental varieties or lines, H19 and G421. First one is monoecious, second one gynoecious. First one has a lot of lateral branches, second one or two. First one has a poor fruit quality, second one good one, and first one has sequential fruiting, and second one non-sequential fruiting. Next slide, please. So from that cross, there was a yield of 171 recombinant inbred lines using different kinds of. Then those lines were planted in two locations in 2 years, and we were looking at those, let's say, most important, let's say, QTLs, and we were first producing the genome map at that time, not too many points, but nevertheless, it represented all 7 linkage groups, 7 chromosomes, and we were able to continue our work with QTL analysis. Next slide, please. So for that one, we have identified 6 QTLs for sex expression, 5 QTLs for multilateral branching, 2 QTLs for earliness, and 5 QTLs for fruit length. And on the right-hand side, we can see the correlation among traits that was actually quite, I say, correlate between each other, and that kind of added the complexity of the exercise that we have started to work on. Next slide, please. So we said that we need to start dealing with multi-trait selection and compare phenotypic and market-assisted selection for those multiple traits, and looking at 5 traits and the use of different marker types for market-assisted selection. And we were as well evaluating 3 cycles of selection to see the gain from selection. And as well to characterize the epistatic interactions among QTLA conditioning, sex expressions, and multilateral branching. So for that, we needed to create near isogenic backcross lines and assess those lines under as well abiotic stress conditions. Next slide, please. So this is once again just what Neus already showed a couple of times, the difference between near isogenic lines development and bulk segregant analysis. This is what kind of combinations what we have used. Next slide, please. And this is the scheme of the backcross near isogenic lines, how it was created with 5 different, let's say, QTLs or populations that were defined by the modifier of the sex expression. Since sex expression in cucumbers it's not like simple gene, but has as well a series of modifiers that is determining their sex expression. Next slide, please. And let's say one of the results when we looked at we have identified QTLs for specific linkage group where we have several QTLs that are sitting on the part of the chromosome that is going to be used or was used as well for further dissection of those traits, and using that then as well in the marker assist selection. So the last example is the drought tolerance in common bean. And this study was aiming to characterize quantitative traits as a differential response mechanism to water limitation in 2 bean cultivars contrasting a water stress tolerance, and then to analyze 82 recombinant inbred lines for polymorphisms with different molecular markers to construct genetic linkage map and of beans, and to use that for further QTL mapping. So we have used 2 varieties. One is Tibor and the other one is Starozagorski Chern. And we have tested them for withholding irrigation. In the middle, you have the graph for the relative water content, where we have looked at the 5th and let's say 9th date of draft, so different stages of the drought. And you can see that they were behaving differently, and as well the values of the relative water content were differently for both. So we said, yeah, this is good sign, so we can start developing the RILs from those 2 varieties. And we have as well, next to the water stress, we were as well looking at the seed and flower count, days to flowering, seed yield, 1 seed mass, water potential, and photosystemic fluorescence. And they were as well scored at different stages of drought. And on the bottom, you see the scheme, how we have produced those F8 RILs generation. Next slide, please. This is the scheme of characterization for the QTL mapping. Since first we have produced genetic map of the common bean based on the F2 population, and then we started to look at we started to characterize the plants for the QTL map in the F8 population. So on the left-hand side are the parental varieties or parents, and on the right-hand side, the recombinant inbred lines. What we have looked at, and next slide, please. Here is the result. What we have then produced, the QTL map of 21 QTLs linked with genetic markers, and that were arranged in 11 linkage groups. And on the left-hand side, it's a similar graph that Neus already showed for the tomatoes and eggplants, where we have one trait and then the number of recombinant inbred lines that are contributing to those QTLs. Next slide, please. And I think that was the end of my examples. And back to you, Neus and Adrian.
Okay, thank you. Then I think the only thing that is left is just the take-home messages, the fast quiz, and the proposed homework. So if you want, the questions for the fast quiz are these ones that you can see in the screen right now. How can new breeding populations be used for organic breeding purposes? Why it is important to maintain the genetic diversity? And which precondition is important for QTL mapping? I leave you 15 minutes, more or less, to answer these questions. Okay, you can send them to Ana Maria, to Adriana, and me. I also copy them now into the chat, just in case any one of you cannot see them. Okay, and I leave you until 5:45. Okay, see you in 10 to 15 minutes. Sure. As some of you have been asking, I'm sharing also the proposed homework. The proposed homework is proposed as short procedure for QTL mapping, just theoretically, of your favorite crop and your favorite trait in this crop. And you have 1 week to send it by email to Ana Maria, Adriana, and me. Okay, I leave you 2 minutes, and I comment the fast quiz and the take-home messages. So let's finish this unit. I received some very interesting answers to all of the questions. Thank you very much for joining us this afternoon and for answering all the questions. So I'm quickly checking your answers. So I think that the second question, the why is important to maintain genetic diversity, is the most clear, because as all of you have understood, maintaining genetic diversity is crucial for developing more resilient materials to be able to propose further breeding projects. How can new breeding populations be used for organic breeding? There are a lot of different answers, and a lot of them are correct. For example, they can be used for widening the genetic diversity of a crop. They are also used for selecting for complex traits and wide the genetic base for complex phenotypes like adaptation to climatic conditions, to organic breeding conditions, or to low input conditions, and in short, to increase and improve the tolerance of the resilience better of the crops. And which precondition is important for QTL mapping? If Vladimir wants to answer better this question better than me.
I think it's the simple one. So it relates as well to either QTL or any kind of mapping. We need the polymorphism. We need to see the differences, and if we see the difference and if we can measure those differences well, then we are all set to start our mapping, let's say, populations, and then as well the mapping procedure.
Thank you very much for this last answer. And just I'm going to give some take-home messages. The first one is that traditional breeding has significant limitations, and we need to have this into our minds, and we need to have this into account because complex traits depends on a lot of genes, and selection and traditional methods have led to less and less genetic variability. So it's important to develop new breeding population that exploits the natural diversity that we already have in our gene banks. And it's just not breeding populations, just combines this diversity that we have to be able to give a new material resources. Also, multi-parent populations can be used for develop tolerant and resilient varieties for organic or low-input agriculture, and a lot of other information. And obtaining new genetic information through these populations is key for a more sustainable development and the easy sustainable development and fast development of new cultivars. So that's all for today. Just remember your proposed homework is also in the chat, and tomorrow we receive this presentation so that you can also prepare your homework. Here you will have in the presentation also some additional resources, some bibliography in this matter. And that's all for today. Thank you all for your attention today, and see you on the module 5 this Friday at 9 a.m.